I think our ability to identify the ideal drug targets is accelerating massively. This is owing not only to multi-omics and our ability to identify this is the absolute key target and this is likely to work in disease and not affect normal tissues, but also in the chemistry of drug development, where we've gone from small molecules and biologics to now so many additional modalities, ADCs, T-cell engagers, radioligand pharmaceuticals, CRISPRs, ASOs, PROTACs, molecular glues. So the armamentarium of the proteins that can be drugged has massively changed, and the weapons we have in which to do that has grown substantially. And so being able to identify the optimal targets now has become very important. Multi-omics enables that in a very meaningful way.
Welcome back to the Smart Biotech Scientist. I'm David Brühlmann, and this is part 2 of our fascinating conversation with Mo Jain, who is the founder and CSO of Sapient. If you missed part 1, we explored how multi-omics technologies are transforming our approach to drug development and disease understanding. In this second part, we will get practical. We'll discover how you can implement multi-omics in your own research, the computational frameworks that make sense of thousands of data points, and the future possibilities as these technologies converge with AI. Mo also shares invaluable advice for scientists like you thinking about commercializing their technologies. Whether you're a bench scientist curious about omics or a team leader considering new analytical approaches, this episode will give you actionable insights. Let's jump back into our conversation with Mo Jain. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. I would like just to dig into a bit more into the technology, because you developed the technology that allows, if I understand correctly, so please correct me if I'm wrong, do all the multi-omics analysis in one single run, right?
Yeah, so that's close.
Or close to that. How do you do that?
Yeah, absolutely. So one of the real fundamental advances that we had helped, and certainly there's many people and many groups that have been involved in this, was being able to take these instruments, these mass spectrometers, and make them go much faster, on the order of 50 to 100 times faster, and to be able to measure many more molecules than would be typical. So the depth of measurement was far greater, as well as the speed at which we do it. And the nearest analogy I can say, David, is every year when you go to your physician's office and you go for your annual physical, they draw those 2 purple tops of blood. And in those 2 purple tops, we measure about 20 different factors. And those 20 different factors is how we make the majority of diagnosis. They tell us about your heart and your kidneys and your lungs and your liver and most of the fundamental processes in your body, and tell us if something has gone wrong. Now, when we think about it, we're measuring only 20 things, and there's 20,000 things circulating in your blood. And how did we pick these 20 things? It's not because they're necessarily the 20 most important. They were just the 20 things that were developed because they were high abundant and we were able to measure them reliably many decades ago. And so what we're doing here is filling in the gap whereby rather than measuring just 20 things, now we're measuring 20,000 things. And this includes thousands of proteins, thousands of metabolites, thousands of lipids, cytokines, other protein isoforms, et cetera, et cetera. So we're able to fill in the gaps here and go from 20 to 20,000. And that's really fundamentally how the technology works, or what it's doing. Now, how it works is in order to do that, you need to increase not only what can be measured at a singular time, and there's a number of innovations that occurred in that process, but also the speed at which it occurs. Measuring 20,000 things, but taking hours on time means that you can never scale this to a large enough population where you can make sense of the underlying data. And so both problems had to be solved simultaneously, and that was the last decade and a half of my life, working with folks who are much brighter than I am, engineers, chemists, et cetera, to bring these technologies to bear. And that was the birth, if you will, of Sapient, was the commercialization of these technologies and these services in order to be able to do this within the context of the biopharma industry.
And what kind of biocomputational framework did you develop? Because obviously, if you generate 20,000 data points or you're analyzing different things, you need to make sense of that and very quickly, especially if it's a routine analysis. So how does that work?
Yeah, it's a great question, David. So we learned a lesson very early on in Sapient's life whereby we would generate these substantial data sets and we would transfer them to large multi-billion-dollar organizations that had dozens of bioinformaticians and biocomputationalists on their teams. And we could almost set our clock for, it was about 7 days, almost to the minute, where we would get an email back saying, what the heck do I do with this? And so we learned very early on that to make use of this data and to recognize the value of it and to really be able to gain the knowledge, again, generating the data was only step one, that we'd have to have services also for biocomputational support, whereby we developed teams of individuals who had deep expertise in the handling of this type of data and subsequently the integration of it in a way that allowed us to be able to derive what we call actionable insight. And that fundamentally means answering a question, a biological question typically, that allows someone to go faster in their drug development process. Now, there's many flavors of that. It could be from what is the best target for this disease to what's the mechanism that drives this disease to how is this drug going to work to how do I screen these thousands of drugs to understand which is the best one to how do I understand the toxicology of this drug? How do I enroll patients? Who's going to respond? Who's not going to respond? So there's many, many, many flavors of biological questions. And we have to have an understanding not only of the data architecture, how that data is generated, the limitations of that data, its structure fundamentally, how it should be handled mathematically, but equally important is to understand the underlying biology. And this is really the field of biocomputation. And so we worked very early on to develop that team of individuals here internally at Sapient that would be able to provide that service. And so we can generate the data, and then we have a whole team of individuals internally that's now frankly larger than our chemist team. It's larger than the folks who are generating the data as a team of individuals who are analyzing that data, again, with an eye towards answering a key biological question.
And where do you see this going, Mo? Because now we live in an exciting time with AI models pretty much surfacing every single day. Obviously, we don't really know where this is taking us. But what is your vision of the future of omics, leveraging all these new technologies?
Yeah, it's a great question. And anyone who knows me knows that I'm an eternal optimist. So I fundamentally believe that the world is changing for the better, despite it may not always feel like that on a day-to-day basis. But the world of medicine is changing for the better. And we're just in the first inning of this. There's much to go. And it's almost like a toilet roll type of analogy whereby as we get into this further and further, it gets faster and faster. The toilet roll goes faster. And so the way I think about this is I think our ability to identify the ideal drug targets is accelerating massively. This is owing not only to multi-omics and our ability to identify this is the absolute key target and this is likely to work in disease and not affect normal tissues, but also in the chemistry of drug development, where we've gone from small molecules and biologics to now so many additional modalities, ADCs, T-cell engagers, radioligand pharmaceuticals, CRISPRs, ASOs, PROTACs, molecular glues. So the armamentarium of the proteins that can be drugged has massively changed, and the weapons we have in which to do that has grown substantially. And so being able to identify the optimal targets now has become very important. Multi-omics enables that in a very meaningful way. Certainly, there's a number of innovations around AI-based chemistry for drug development. Multi-omics allows us to take these AI-derived agents and very quickly screen them to understand how they're going to work, what their primary effects may be, what their secondary effects may be, what their adverse events may be. And then fundamentally, I think where we're going to see the greatest amount of innovation is in the diagnosis of disease. which again, really fundamentally hasn't changed in several decades. But the way we think about disease risk and diagnosis is going to be changed pretty massively, I suspect, in the next decade. And then how we select who's going to receive a particular agent. And so what I mean by that, and we talk about this as personalization of therapy. And when personalization of therapy first was introduced, the concept was that the differences between you and me, David, are what derive our disease pathology, and subsequently orienting a drug that's best for you or best for me is the key. I think that's evolved quite a bit, in which we realize that you and I are actually pretty similar, but our disease may be fundamentally different. We may say that we both have diabetes or cancer or Alzheimer's, but that really represents a spectrum of diseases, and understanding how your specific disease is different than my specific disease is really the key. And this is where multi-omics implementation as part of personalization, I think, is going to change massively. We've already seen this in oncology, and I think, again, we're just in the first inning of this.
To what extent will these technologies enable us to detect diseases much earlier? Because I think that's also where we need to go, because oftentimes we just find the disease too late and it's too hard to treat, right?
Yeah, you're absolutely right. So if we look for most chronic conditions over the last 50 years and see where the greatest change in mortality has occurred, the answer is very clear. I mean, it segments into two areas. For acute diseases, it's antibiotics and septic treatments and ICU care, right? That's really what's given rise to the majority of mortality benefit over the last 50, 60 years, 70 years of medicine. If we look in chronic conditions, it's almost always an early diagnosis. So it's much easier to fix the car if you can catch it early on before, when it's just starting to break, or even before it's breaking, as opposed to after the wreck. And that's just the way medicine in the body is. After the wreck occurs, it's hard to fix it and put it back together. You can mitigate it, but to really improve mortality and meaningful health span, you have to diagnose disease early. I go back to the analogy that I used earlier, where the majority of diagnosis, at least from a laboratory perspective, is based upon a couple dozen things we measure in blood. And there are tens of thousands of things that are in blood. Being able to now to access that information stream very reliably in a robust way, and then make use of advanced computational algorithms, the AI that you referred to, in a way that allow us to do much, much better disease prediction. I think this is going to fundamentally change medicine, and again, will allow us to diagnose earlier, to better understand prognosis, and then fundamentally to much better align an individual's disease process with a particular therapeutic.
Now, let's make this very practical to a biotech scientist who is on the fence, who thinks, well, I would like to do some omics studies. What is your advice? How should they get started?
Yeah, it's actually quite simple. The way I always approach this is forget what you're going to do. Start with the why you're doing it. What's the question? What are you trying to answer? Are you trying to identify the best target? Are you trying to understand what may be the potential on-target, off-tissue adverse events related to a particular therapeutic? Are you trying to understand who's going to respond to a therapeutic or not? And so really this again comes from fundamental biology and medicine and understanding, what question am I trying to answer? Once you have that information, this is when then you can begin to layer on the sort of ideal data modalities. And I really don't believe in the many nails, there's always one hammer, or if you're a barber, everyone needs a haircut type of approach. But rather, this is about understanding the technologies, understanding the data, and again, having a sense as to where that signal may lie. And this is what we fundamentally do here at Sapient, as well as other organizations. We work with our clients. We're a service organization. So our job is to take individuals who have those clear questions, who are developing therapeutics, and to help align them in a way in which we can provide advice regarding potential omics, whether it be a singular omics, whether it be multi-omics, and then to help them design the ideal experiment that's set up to answer that question, and then to go about executing on that. Again, generating that data, QCing that data, doing the statistical analysis of that data, fundamentally answering the question for them, and then helping them advance that information forward through their drug development pipeline.
And any pitfalls to avoid or to watch out for?
Oh, no, of course. This always works. Yeah. Yeah, absolutely. I mean, as with anything, I hope I'm not oversimplifying it, but there's a number of pitfalls. First, biology is incredibly complicated. Medicine is incredibly complicated. It's remarkable when you really think about the process of drug development and what we know and what we don't know. It's really remarkable that this ever works. Ever works. I'm still shocked that we are able to develop drugs ever and that they have any benefit. And I can't tell if it's just sometimes we get lucky, but humility is a core component for anyone doing science. So yes, there's tons of pitfalls, and it can be as simple as the information doesn't lie there. We simply can't predict X, Y, Z, or the system doesn't allow us to understand what the ideal target is, or there's so many technical potential pitfalls. There are so many biological pitfalls. There's so many practical pitfalls. How do you take this information stream and how do you turn it into clinical tests? And so there's many, many, many pitfalls along the line. But what I can say here, David, is that the juice is fundamentally worth the squeeze. And so despite the pitfalls, I'm shocked, and despite my optimism, I should say, I'm shocked how well this does work and how many times we say, wow, this was unexpected, but this is really the answer. And the data is telling us the answer is something a little bit different than we thought it was going in.
Now, as we're getting close to the end, I'd just like to circle back to the beginning. Coming back to your career, you moved from academia now into a leadership position, founding your company. What is the number one leadership advice you would give to a scientist who is also thinking of spinning out his technology into a company?
Oh my goodness. That's a tough one, David. I'm not sure that I have any right to ever give anyone advice. What I can say is that, well, there's two pieces of advice, one for the organization and one for the person. One, you have to be very humble to do this. And what I mean by that is that there's almost no meaningful company that I'm aware of that's been developed and built in a linear fashion. All of these are very circuitous. You will make many, many, many more mistakes than you'll make right calls. The key is to learn as quickly as you can from those mistakes and do everything you possibly can to not repeat them, and to make sure that you have the emotional wherewithal, not the intellect, but the emotional wherewithal and the support system that allows you to wake up every day and make mistakes and get up the next day. And that's tough. That's not an easy prospect under the best of circumstances, but that's an unforeseen challenge that became extremely important to me personally. And then from an organization perspective, as you know, David, we heavily are invested in technology development and data sciences, et cetera, et cetera. And I fundamentally believe that none of that makes a difference in the end. The best technologies are just paperweights. The best algorithmic work is just a series of zeros and one on a hard drive, unless you have the right people that can turn that into magic. And you hear many organizations say it's all about the people, but I really, truly believe that. If you don't have the right team, if you don't have the right individuals, you can have the world's most groundbreaking technology. You fundamentally won't be able to do anything with it. And it just takes up space and weight. And so really investing heavily in the right individuals. What I would say is that the world has changed because of computing and other aspects and technology, and that you don't necessarily need 500 people or even 100 people. You could do remarkable things now with dozens of people, whereas just several years ago, that took hundreds of people, and before that, thousands of people. And so I think that's the big sort of evolution here. You can be much more selective as to who is part of that team. But in the end, the team is everything.
This has been fantastic, Mo. With all what we've discussed today, what is the one single takeaway you want to walk us away with?
Yeah, I'll go back to the first question that you asked me. Well, what's sort of an unconventional truth that you believe? And that is that drug development is really a broken process that, again, I'm shocked ever works. And what I would say now, we're at a dawn of a new era whereby I fundamentally think we are primed, given the types of data that can be generated, given advanced computing, given the acceleration in our ability to understand information and derive knowledge, that we're going to see real accelerations in drug development. And that's extremely exciting to me. I almost feel like this is at the dawn of antibiotics or the dawn of medicine, where needles are going to move very quickly. And that's exciting.
Mo, tell us where people can get a hold of you and check out more about omics.
Absolutely, David. So we're certainly online at www.sapient, sapient.bio. I would really welcome anyone to look us up online. Our contact information is on there. We have a bunch of webinars and white sheets and other information streams for individuals. We're also on social media, as you can imagine. There's a ton of information we post on LinkedIn and other areas. You can look under sapient.bio. You can look under myself. And certainly our job, in the same way it is to generate data and to analyze it, is to educate scientists and people who are doing the hard work of drug development. And so we really would be happy to chat with anyone, whether or not they're interested in services or just interested in chatting. Please reach out and we'd be happy to have those conversations.
Smart biotech scientists, use this opportunity to reach out to Mo and his team. I will leave all the information in the show notes. And thank you so much, Mo, for sharing your passion, your expertise, your insights. I've greatly enjoyed our conversation.
Thank you, David. It's been a real pleasure. Appreciate you.
And that concludes our two-part conversation with Mo Jain on Multi-Omics technologies. Mo's insights remind us that we're truly at the dawn of a new era in drug development, one where we can finally make sense of the complex biological data that was previously hidden from us. Remember Mo's key takeaway. While traditional drug development has been a flawed and inefficient system, we now have the tools to dramatically accelerate our understanding of disease and create more targeted therapies. As he put it, the juice is fundamentally worth the squeeze. If you're interested in learning more about how to leverage multi-omics in your development projects, I'm offering a free consultation to answer your questions. Whether you're curious about experimental design, data analysis, or practical implementation, I'm here to help you navigate this exciting frontier. Visit BrühlmannConsulting.com/call to schedule your consultation today. Thank you for listening to the Smart Biotech Scientist. If you found this episode valuable, please leave a review and share it with a colleague who might benefit. Until next time, I'm David Brühlmann, helping you do biotech the smart way. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.